Yeah, that's always good. All right, I heard Princeton join us, so we'll go ahead and get started. It's my job today to announce a few graduate student awards. And unfortunately, Mark Coyne, who normally does this for us as our Director of Graduate Studies, I think he's at graduation. And where was he going to be? I think he was at graduation today. So he put me, and Jason will join me here briefly. The first one up is the Peaslee Award. Periodically, since 1991, the Department of Plant and Soil Sciences has given the Doyle Peaslee Award to an outstanding graduate student in agronomy. The basis for the award is high GREs, good grades, research strength, and potential for contributions to U.S. agriculture. The award consists of a small honorarium and an engraved plate with the student's name posted in the department, which is down the hall here. This year's award winner is described by his nominator as having been on the very cutting edge of soil toxicology and agricultural biotechnology. He has a paper accepted in Environmental Science and Technology, which is considered by many to be the flagship journal of environmental science. He is also described as having dedication to student government and the IPSS Graduate Student Association and Graduate Student Congress. Instrumental in organizing professional development trips for the IPSS GSA and implementing other GSA activities. Please join me in congratulating our winner of the 2018-2019 Doyle Peaslee Award, Stuart Lichtenberg. Come on up. Yep, come on up. We're going to take a photo. Jason, if you want to come on up. At least I think, oh yeah, there she is. Next on the list is the Gerald Mott Award. The Gerald Mott Meritorious Graduate Student Award in Crop Science recognizes top-notch graduate students pursuing advanced degrees in crop science disciplines. Departments select students based on academic achievements, research and teaching contributions, leadership accomplishments, service activities, and personal qualifications. The awards offered by the Crop Science Society of America, and recipients receive a certificate and are recognized in CSA News. The award honors the first CSSA president, Gerald Mott, who trained 75 graduate students during his 45-year career at Purdue University and the University of Florida. This year's winner is described by his nominator as conducting research on Fusarium head blight of great significance in Kentucky, in which wheat is a central component of traditional rotation systems. Fusarium head blight poses a significant problem to wheat producers due to decreased yield and the value of grain caused by production of the harmful mycotoxin DON. The winner is making fundamental advancements in basic plant breeding on a problem of significant economic consequences. His dissertation work will be of great benefit to the Commonwealth and regionally. Please join me in congratulating our winner of the 2018-2019 Gerald Mott Award, Jesse Carmack. Okay, and then starting this year, the IPSS graduate program has tried to recognize outstanding new and continuing students who demonstrate high performance in knowledge, skills, and communication and professionalism. You all may know this was something Mark started with our IPSS block funding this year, and you did it with your annual, basically the grad student annual reviews. Our 2018-2019 winners fall into several categories, and we'll start with the best new PhD students. They awarded 2 in this category and in the best continuing PhD student because there were no nominees in the best new master student. So this year, our second place best new PhD student is Elena Jacobs, and our first place winner is Dan Quinn. Come on up. Next is our best continuing PhD students, and again, 2 winners. Second place is Elisiane Weber-Tessman, and first place is Catherine Rott. Yeah, right, graduation. And the last one for today, before we start with our speaker, is the best continuing master's student, and that goes to Cynthia Schiaruzzi. Okay, and now I'm going to turn it over for Art for an introduction of today's distinguished alum.
Okay, thanks very much, and congratulations all the students. So today it's a great pleasure of mine to introduce the 2019 Distinguished Alumnus Awardee for the Department of Plant and Soil Sciences, Dr. Dongmei Zhu. So Dongmei is a native of China, so she earned her bachelor's degrees and master's degree from Shanxi Agricultural University. So Shanxi is like halfway between the Terracotta Warriors and the Forbidden City. After earning a master's degree, she joined the Department of Agronomy in the crop science PhD program here at the University of Kentucky. She joined Glenn Collins' lab, worked with Glenn Collins and Mark Nielsen to do tobacco breeding and biotechnology. Earned her PhD, I think, in 1996. Thereafter, she stayed with Mark Nielsen, moved to I guess a startup company, and ultimately out to Winchester, Kentucky, working for U.S. Smokeless Tobacco, where she continued to develop as a very accomplished research scientist in doing tobacco biotechnology. While she was there, she also served as an adjunct faculty member in the Department of Agronomy for several years, served on several graduate student committees. I think 2008 or 2009, she moved from Winchester to Altria Client Services in Richmond, Virginia, where she moved, I guess, through the ranks. So she joined as a principal scientist, moved up to become a senior principal scientist, and recently she was awarded the rank or whatever as fellow at Altria Client Services. She's had a very distinguished and productive career in research and administration at Altria and also at U.S. Smokeless Tobacco. Her list of accomplishments really would take too long to even start to mention. I would just say that one area that I think we probably most very familiar with and in which she's been really a pioneer is the development of tobacco varieties or lines that have very low nornicotine production, and so in the area of developing tobacco lines that are going to be lower in carcinogens, she's been a pioneer, an absolute pioneer in the field. She's involved with many aspects of research, and she's actually going to talk about them, so I don't have to read them all off this paper that I've got here. So Dongmei has published numerous peer-reviewed research articles. She's kept active in publishing over the years. She's a named inventor on some 20 or more United States patents and 100 or so international patents. She's internationally recognized as a leader, maybe so one of the leaders in tobacco biotechnology research. She's currently the president of the Phytopathology and Genetics Study Group for Caresta. I could go on and on, but rather than do this, I think I will turn to her nominator for this award, Dr. Pfeiffer, who explained more succinctly than I can the impact that Dongmei has had on her field. So Todd said, Dr. Zhu is a highly regarded research scientist whose numerous scientific and technical contributions have benefited her company and the tobacco industry in general. Her research in gene discovery, tobacco trait improvement, extending product shelf life, and identifying genetic mechanisms for tobacco harm reduction have placed her company's R&D program in the forefront of the tobacco industry. Dongmei's elevation to a fellow is a rare distinction at Allotetraploid Client Services. It is only bestowed on scientists who make major contributions to the company's objectives, have achieved national and international recognition for their research, and are considered expert in their field. Dr. Zhu continues to provide creative scientific leadership in a challenging environment. She is truly an outstanding alumnus of our graduate program. I introduce Dr. Zhu.
So I think I got it. I'm so glad to be back to my home school, and I'm so happy. I have so much fun here 20 years ago. So it's my great honor to receive this award. So I didn't know I will have opportunity to speak more or not, so I want to thank a lot of people. So first, thank my family for everlasting unconditional support. Then mostly, I will thank University of Kentucky, College of Agriculture, and our own department provided me a golden opportunity to pursue my career. And most specifically, I will thank a lot of faculties, professors, and the staff who helped me going through my study process. Then learned not only knowledge, and also how to become a good scientist, also how to become a good person in whole. So this, first I start from Dr. Pfeiffer, who has faith on me, have got, dare to brought me from all the way from China to here. He's the first professor, and the second, Dr. Mark Nielsen and Glenn Collins provided me opportunity to pursue my PhD degree, not only provide me financial support, and also guidance on the patient, everything went through, put up with me. Then Dr. Arthur Hunt guided me in the molecular biology. I start from nothing, then I learned the skills from his lab, and also his faculty staff member from Carroll. So I learned a lot from them. And the next group are Dr. Egley, Dr. Wagner, Dr. Pierce, Dr. Bush, then helped me a lot along my career. Then not only for my study time, and then later we become two friends and as mentor along my career. And also Dr. Dr. Barrett, and to allow me to work for University of Kentucky for many years from our department as an adjunct faculty. Sorry, Dr. Barrett. And so I have so many more people to thank, but without overdue, then I just remember one example. A few weeks ago, I was sitting in a panel discussion in U.S. Patent and the Trademark Office in Washington, D.C. It is called Newman Court, in of the court discussion. It's regarding women in science, women innovator. They asked me, during your career, when and where is that important time or most impressed time or occasion to shape your future or to drive you into science. I have no doubt in my mind, University of Kentucky. So all professors' dedication, passion to the science, then the capability, knowledge, and also drive lead me to where I am. So some example, Dr. Nielsen in field, day in, day out. Then I work along with him. I have 13 field trial for my thesis. Not exaggerated, it is true. And he worked with me day in, day out along on his research. If I one day don't go there, miss the virus incidence record, I feel ashamed. So also the professors have a lot of patience on me. Dr. Joe Chappelle. Then when I take his molecular biology class, first quiz I got is lowest grade, lowest score I never had in my life. So he put me into his office and tell me, let me give you some introduction book. So basically molecular biology 101. So he gave me a long list. I read it like crazy, and end up I got an A. So that's quite experience, and I can go on and on. So I will stop right there. Thank you for committee selecting me as this winner, and also thank you for all of you, your support. Without your support, I will not be where I am, and my family will not be where we are. I'm honest, really, I'm emotional. And for that part, a lot of people help my family. And so I'm going to, today I thought a lot of high-ranking professor here, I feel humble. So I'm not, instead I dive into science deep. So I'm going to just introduce how biotech program works in a corporate environment, how to connect the science to the business solution. So with that, I will start. So title of my talk will be Tobacco Trade Development through Biotechnology. So I'm going to touch this, talk about the structure of a biotech organization, at least my group, and also move on to what platform technologies we have so far on top of the hard infrastructure. This will include the tobacco genome database, and the omics, the platform, and also we newly made the Axillary chip. Then I will move on how those genome information or modern technology move the needle of the traditional breeding, and how to know what the natural variation of the germplasms, what's the difference, and what is diverse or not diverse, or what is novel traits there we have possibility to get it. So finally, I'm going to touch 3 examples. How do we develop a trait? So this will be a very low nicotine, very low alkaloid tobacco line development, and the nitrogen utilization efficiency, and sucker control. So first, I bring us to a little bit of genetic background of Nicotiana tabacum. So million years ago, 2 wild species, Nicotiana sylvestris, Nicotiana tomatosiformis, they got together, made a random hybridization. So our commercial tobacco is evolved from that. So what that means is tetraploid. So genome complex, genome content is so complex. So if you look into the chromosome level or genome level, you can see the genome size is big, 4.5 gigabit. It's way bigger than the human genome, way bigger than soybean, corn, tomato, rice, and so on. Then also genetic makeup is so complicated. A lot of repetitive genes. Some of them, they are treated like diploid, and some of them is indeed tetraploid, or sometimes they have a lot of analog inside. So it cost me a lot of trouble because my career is on tobacco. And So since it's so complicated, also our program moved to a so-called modern phase. So with the development of the sequencing technology advancement, so we choose one common tobacco, Burley tobacco, Tennessee 90, which developed from our professor, Bob Miller. So we use that as a reference tobacco, reference material to thoroughly genome sequence them. So we want, with timing, we want many, many generations, start from back library tagging, which is very classical. Then we go to Illumina sequence, then we go to 454, then we go to PacBio, then we go to Dovetail. Finally, we connect all those colleagues together, become to 24 of the pseudo molecule, which match the chromosome. So right now, if we have a trait, if you have a gene, even QTL, you know what the genetic makeup a little bit. If I search on my database, I will know belong to which chromosome and connect with gene. So it's not all of them, but most of them. So after that, we also get 10 reference genome, did a resequence onto it because we want to see the diversity. So those resequence means you just use the reference genome, the sequence information as a template, then you find out the single nuclear polymorphism throughout the entire genome. So with this 11 lines, we generate hundreds of thousands of SNPs. But through the data curation and also purposely chosen, then we selected 170K SNPs cross over the genome, then we made the Axiom chip. So from then on, this chip with a server base to accelerate our breeding program dramatically. I will tell you some living example. Then with other omics, we move the needle of the trait discovery. So like I promised, I will introduce you a little bit our instructor, how our infrastructure is, how our organization organized. So as a biotech program, on top of the microbiology others, in plant set, we have 4 groups. One is gene discovery group, another one is the bioanalysis group. So these 2 groups get data, provide insight, have insight, then you develop trait from it. Then a breeding group is you can continue doing convention breeding. On top of that, we will transfer the trait developed from this discovery group into other varieties. Name it, popular variety in tobacco right now is around 30, 20, 30 across all different types. I will explain to you later. Then for the platform, besides traditional breeding, we do have genome information database. I already said that. Then we have RNA-seq, that means transcriptome, proteomics, metabolome, and chip. All those pipeline is in place. So how do we evaluate the function? We have tissue culture system, start from protoplast, cell culture, and the normal callus tissue culture. So we can do different level for different purpose to evaluate the function of the gene. Then we also have gene editing tool on the EMS mutant genesis on the screening pipeline to develop a non-GMO commercial line. Then for breeding, in the little center squeeze into, then the first one, we have FT technology, which can short dramatically of the tobacco growth site. So some example case is only 1 month, 2 months, you get a seed. Then we have marker assisted front selection. Then we have gene chip background selection to speed up the less cycle to get final lines. For evaluation, we have, of course, everybody knows this, have greenhouse, experimental station, then also set the nursery. So southern nursery is special for us, can speed up. Every year we can do 2.5 season cycle or even 3 cycle. So that's our infrastructure on the plus the pipeline. So what we are doing, why we need those, basically is deliver business solution. So we're, forgive me, it's a tobacco manufacturer company. So what you need is good leaf. That means here, good leaf to put into product. So this good leaf means the yield, quality, tolerance, disease resistance, and special attributes. So how do we get there? We use the traditional breeding, marker-assisted breeding, and use all the omics and the gene discovery tool to find the gene even not exist there. Then we use the gene delivery and gene editing tools to deliver. So casing is to get this thing. And if you use those gene discovery, gene delivery to get one line, probably this line possibly is not a commercially best one, then we use through this breeding process to expand to all good varieties. So that's what we are doing currently, nutshell. So now I move on to application on the breeding with this so-called genome technology. So first, I mentioned the traditional breeding still is our core backbone. So how do we do it? Everybody knows this is agriculture department. Do crosses with 2 lines. One has special trait as donor, one good lines we call recurrent parent. Then we do the cross. Then here, no difference. However, if you start to do the back cross, so now the genetic markers or technology play a role. First, you use your marker assistant trait to select Dan in this group is doing that, to select the gene of interest. So you even don't need to go to field. You know this trait exists at the segregation by marker to tag it. So second, you use the gene chip to look into the back cross. The back, we call front selection, back selection. That means how many percentage of the elite lines genetic makeup recovered from this single plant. So when you do the back cross, then you can have list of segregation population, right? So you choose best one, like here, and you continue select it. Third generation, you can reach to recovery as 99.9. So famous breeders are there. Then I hope I was right. Need an 8th generation to get there if you just do the phenotype selection. On top of that, we don't need to get a real world every single time to select the phenotype, to select the best one. Then you only need a last few generation or 2 years evaluation. So you speed up the process tremendously. So a moment ago, Dr. Hunt talked about this trait. I worked on with Dr. Nielsen. Start from this, find this, discover this gene, then I just fast forward, made the mutation, then we fast forward. Then we generate a lot of, expand this trait with 3 separate genes in 3 separate locations, put it together. Then we use this method I said, you can get one single line 3 years to 4 years. So we expand all to the dark tobacco, that's our butter on the bread on the butter. Then we also expand this to Burley and the flu cured. Right now, this material can reduce 70% of the nitrosamine, crossboard, also in product. So we generate 1 million pounds of this put into product. Then this FDA proposed a standard, want us to cut this to certain level. So we are told to at least prepare to meet that requirement, which started from many years ago. And that's the example. So let's move on how to characterize the variation through the through the genome. So first, we can characterize genetics of key commercial germplasm. You know what makes this line superior, or what makes this line unique. I will share with you momentarily. And also you can identify exotic genetic information variation from key breeding block. You even probably use exotic germplasm or even non-tobacco like Nicotiana, wild species, even tomato. So use genetically, you can transfer like a not existing gene disease resistant into tobacco. Those are just a statement. So you will see some example. So come back, we look into the variation. So a moment ago, remember we did 11 lines. So now we, based on calculation, they said the 48 lines will cover entire different type or different unique germplasm diversity. So we listened to the literature, we did it. So we select the 48 lines, we did gene chip, use our famous chip, hybridization. So generate tons of SNPs. So we look into the variation about the germplasm. So if you look into color, just mainly yellow, pink, blue, green. Those you say automatically just do a SNP cluster. You can see they cluster to different types. But not only that, the resolution is higher than that. Look, that the red portion, that's the Burley tobacco come from international source. And the one have the difference, that's the cross lines between Burley, Maryland, and others. So we can tell it. Then especially middle portion, this portion, those are Maryland cross with Tennessee 90 for a specific trait. Then you can see in the segregation population, the recovery rate from Burley component are different. So you purposely choose this one, follow on, do the back cross, or do the cross, then you will get higher one. So even just from this, you have illustration. Another important thing we found out is, unfortunately, the genetic diversity in tobacco, common tobacco germplasm, it's very narrow. So that gave us, this is not only our conclusion, and Ramsey Lewis, Bob Miller, all talk about this, especially Ramsey Lewis through the SSR, all those diversity analysis. So we got a little bit, as a breeder, we got a little bit stressed, sometimes put me into not sleep. One day, if a devastating disease come over, tobacco might wipe out. So that's we need to do for the supply chain security. We need to continue to do more, right? So this is a modern technology I'm doing right now. We do whole genome breeding. What you do is you collect all the germplasm you feel interested, use different purpose, then you shallow sequence it. That means you get the SNPs or even more sequencing information. You use the high bioinformatics tool to cluster them together. You can form this haplotype block. What does that mean is you can analyze this individual block, find out which block is related to, for example, this one is related to evolution. And this block is related to the bread on the butt or bread butter of the tobacco. So they have to have this. This is essential, so no variation. But here is the interesting part. You can see some trait gain during the evolution or during breeding, or some trait lost. So you look into again, you can dissect all the piece of it to look into the trait you are interested in. Condition is your phenotyping has to be fit this genotyping. If you don't know the phenotype you have, you don't have this correlation, you cannot do anything with it. So then use this way, some people already did a lot to find out some unique gene. For example, what is broccoli, cabbage? What's the unique gene between? Then they find, actually, I'm sorry, broccoli and the cauliflower. Then they already have things move on. So we are doing that for tobacco flavor, tobacco quality, because those are critical things for us from leaf, not from grain. So this is an illustration of application with timing limit. So I'm going to move on to my last portion, but it's lengthy. So trade development. Again, we look into the business needs. So in tobacco world, on top of those product quality, disease, routine demand, we have upcoming even more stringent demand is FDA regulation. Last year, FDA announced ANPRM proposed nicotine level potentially will decrease to 99.9% reduction, reach to 0.4 milligram per gram. So very rather stringent, right? So meanwhile, while we working with them to see what is appropriate level, but we also need to actively prepare for it, right? So here is the strategy in my lab I'm working on. Besides the routine breeding, I'm going to talk about a little bit, then we has additional many strategies. This is 2 examples. Target gene knockdown, then blocking transporter. So first one, this has to accredited by back to University of Kentucky, Dr. Pollack and Dr. Glenn Collins developed low alkaloid series back to 1950s. So those are 3 lines, normal, and they have 2 locus, big locus deleted by crossing with cigar tobacco. So if you have NIC2 deleted, it's less impact, you have high intermediate, 30% reduction. Then if you have NIC1 deleted, which has more impact, then you will have 70% reduction. If you have both deleted, you can have 95 reduction. We take use of this material, I think all world people are using, which is again incredible back to University of Kentucky. However, a serious issue. Leaf quality is bad because one is big chunk of deletion, right? Second is we don't know correlated with low alkaloid or not. So look at this is normal leaf. I am non-tobacco people probably will feel, oh, this is yellow, but the green is the bad one. We need this richness of yellow and have flavor, everything. But this one is clean, flashy green. If you put into a curing room, later on become to a piece of brown paper or greenish paper. So it didn't work. So we want to, our objective is to develop very low nicotine lines that maintain this good leaf quality. So then forgive me, I need to get back deep a little bit here. So nicotine contains pyridine, 6-ring pyridine, 5-ring pyridine. So if you look into biosynthesis pathway, they have 2 pathways too. Left side start from aspartate, then you go to the quinoline, go to QPT, then you get a 6-ring structure. Then the right side start from methionine, and the only thing goes through ODC, ADC pathway. Then you through PMT you get a 5-ring part. Then they combine together, become to nicotine. Of course, other metal alkaloids and MAC there too. Then also no nicotine is the demethylated product of nicotine. So I just illustrated 2 examples. We block the key enzyme or enzymes list of PMT to look into what is the performance. The next one is PR50 is a regulated gene. So if you look into, I pass through all the gene discovery, blah, blah, then RNAi reduction results, these are RNA, downregulation. So if you look into the line, those are normal tobacco nicotine level. Then those are one of the PR50 RNAi line. This is another one. This is the PMT. And this is that low alkaloid Burley 21. So they can indeed downregulate to 95. However, the quality again is bad. That is low alkaloid Burley tobacco. But for our new developed one, improved a little bit, move needle a little bit. So we did smoke everything. It's better than the low alkaloid, definitely. So that's the preliminary result from that side. Then you look into detail, then you already know the nicotine. Then you look into the leaf quality, and that you can tell whether for the right side, yellow one, that is the PR50, then the PMT, both look better than the second one, which is low alkaloid. You see that this greenish yellowing also thread like a paper or saggy cloth. and this one has some body, right? So that's quality. Now we look into the detail. Why is that? So quickly, very quick. So we did the gene expression analysis. You see the if normal tobacco compared with low alkaloid tobacco, they have 2,000 of gene knockdown, the differentially expressed. But for our other two targets, only have around 20-25 genes difference. So if you look into the PMT, again, reflect the genome, inside the tobacco have 5 functional PMT genes. So how much the headache we have. And that's the normal, 5 gene expressed like that. Then if you go to the PR50, knockdown 3, then low alkaloid, knockdown 2, then the PMT real RNAi knockdown 4. So that gave you a kind of information what the low alkaloid line will be look like. So I'm going to state the FDA's regulation so far is not technically achievable. And I have several people support me, and Jack and Colin Fisher are there. So it's especially commercially, it's not achievable. So now we move this to the non-commercial line, non-GMO commercial line as well. So second is blocking transporter, another strategy, quickly. Then as you know, the challenge is a high alkaloid, high quality, low alkaloid, low quality. However, alkaloid is produced from the root. If you block the transport from root to leaf, to see what happens. So it will limit metabolic consequences or will increase quality. Let's see. So we did tons of external search, find out the wild species, Nicotiana alata, Nicotiana autophora, indeed produce nicotine in the root, but they don't transport to the upper. Super, we can have this to do it, right? To do the comparison. So we have right material work on through all the genetic or molecular biology method, especially on protein. We isolate out the membrane protein from the root, then did a comparison with this normal and the alata. So because transport potentially will be membrane binding, right? So we did that, then along with the RNA-6 data, we select 10 or so of the target. So this is an evaluation of target. So instead of doing real world alkaloid measurement, we did a fading test. So your seedling of transgenic lines knocked down, you use nicotine to see the transport or not. So that's the quick assay we do. Indeed, one of the 5 targets, we found out, you see red is leaf, red is root, blue is leaf. So nicotine, they didn't transport, or transport less at least. So we found those, then we look into the field trial. Indeed, those lines look normal. But we don't know is because is alkaloid still there, have something on the leaf, or is the really indeed the different mechanism cause this. So we are still vigorously working on this. So that will be very low nicotine. Do you have any questions so far? So let me move to the next one quickly. So nitrogen use efficiency tobacco. I forgot to mention one thing. From our organization, we have a lot of work internally, but we extend our arm very far. We have a lot of collaborators even in this room. So we have so many different areas. This is one of the example. Actually, Colin Fisher helped us, and also Dr. Pierce helped us as well. And so then I have other international collaborators, national collaborators do different things because we cannot do everything ourselves. And everybody is familiar with tobacco, so I'm just going to go quickly. Burley tobacco use most of the nitrogen fertilizer. So basically it's a waste crop. They use a lot extra. Then, but however, we found the Maryland tobacco, which is not popularly used in the tobacco industry, but they genetically similar, but they use way less, can produce same level of the same quality, or I would say quality, same quality of leaf from the look and from the yield. Of course, every type has uniqueness. So again, we look into comparison. We did all those things. This is our routine. Then we try to find the solution. But first, I want to show you grafting work, which is striking. So if you do the graph, you put these two together in early stage, then they grow bigger, then you see what happened. So here, and this is Maryland, Tennessee 90 with their own roots. If you swap it, the result come up same. So if you look into data, you measure chlorophyll, you see, you look into cyan, just MMTT. Maryland, top, green. Tennessee 90, top, yellow. So tell us components is in the leaf, not in the root. How can we know that? Then we did a differential expression of the transcription factor. You can see this portion, the major difference is in the leaf-specific factors. But for Burley unique trait, as a comparison, you can see it's in the root on the leaf both. So that gave us, oh, if you keep looking the leaf phenotype, also keep tracking the leaf-related, leaf-related marker, you will get there. The fact, yes. So we did, first, we also do the genetic engineer thing, but let's look into the cross. So we crossed F1 then to F2, then we look into the population, we look into agronomic performance on the marker. So you can see the segregation. However, this segregation not really correlated to the yield. So we have to both, that's another success/failure example. Then some trait you have to go to real world. So that's Dr. Colin Fisher's help come to play. So we are planning to spin the top to evaluate it. So now we get this line, look, have decent attributes, then only need 45 to 60 pounds per acre of the nitrogen. What that means? We will save farmers money effectively, dramatically. 45 versus 118, compare how much we can save. The second is inside the leaf have less nitrogen, less TSNA, which is carcinogen compound for tobacco. So we both do same direction, both go right direction. So let's transport quickly. Sucker control, I still have 10 minutes. Okay, sucker control. Sucker, everybody know this is called sucker. On the Axillary bud, then they grew up like fast, like a tomato. Then we use leaf, not use fruit or seed. So up to this time, we have to top it, remove the top, make this nutrition flush back to the leaf, make it thick. Then the normal mature, you guys already know this. So if one plant without the sucker, then 1 week, you need to remove that much sucker from it. So right now, in dark tobacco, most are removing manually. Poor those immigration laborers have to go there and do this all the time. Then also, for flu and the Burley, do a lot of chemicals. So Dr. Pierce is the king of that. And if you do that, we did a calculation. Worldwide, every year costs close to $1 billion of the money to remove this only. So if we have it, it will be a winner. At least good for our client, right? So what we start doing it is we use this. First, we find the genes, turn on, turn off after talking. Then the second is we find a bud-specific promoter, then combine the promoter with a certain targeted gene, then we can make it happen, right? If it's not possible, we can do tandem expression multiple genes. So that's our first one of the strategy. We also use the laser capture microdissection system to look into the detailed location the RNA-seq to see what gene after topping inside in this meristem portion, primordial portion, what happened, what gene upregulated, what downregulated, right? So we get, this is just an illustration. You see some sample, we take it after topping, before topping, after topping 2 hours. 6 hours after topping, 24, 72. Then we also take a sucker portion, main leaf, main stem, and the root to see the contrast. So we found some specific expression in axillary bud. With the timing, they increase. Then some of them during this time constant. So present there, sucker grew. If you get away, possibly sucker will not grow. We don't know. So then those are lab together as a gene. We need to do one by one as a knockdown, overexpression, to say really have effect or not. So this is one example. If you have this increased gene, knock it down, then you see after topping, then the red panel, sucker doesn't grow dramatically anymore. So some other interesting thing, this is interesting. If you have an inhibitor gene expressed in the beginning constantly, if you remove that inhibitor gene, you see the sucker grow out everywhere. Yeah. So how do we use it? That's remaining. We will do in the future. So right now we just do one strategy. So we find this instead of gene, we use this promoter. And Dr. Joe Chappelle, we did a long time ago about the pathogen resistance, similar. If you use a promoter, it's tissue specific, right? Then you can do something else. I'll explain to you. We put this, we found this 305 different promoter. We put into GUS. Those are expression pattern, different one. So we choose P15 from others, compare. So with time sequence, you can see after topping, the blue spot get darker and darker and darker. So that means looks like sucker specific. So we hook with this with a cell death gene to say what they can do. So if they turn on, cell death gene express the protein, then it will kill sucker. In fact, it indeed do the job. Look, the wild type, then that's P15 with cell death gene. They even do not have primordium in axillary bud. There is a hole in the corner. Then after you top it, then this one grew, those do not grow. Beautiful, right? However, then we found out they cannot pass seed germination. This promoter is leaky. So this is the GUS, compare the GUS the GUS expressed the gene. You see it also the black or blue point. So that means the gene expressed and then cannot pass seed. So that promoter is failed. However, we search other, so this one manage some seed can pass it, pass the next generation. So here's the result. T1 field evaluation. We evaluate the phenotype now. You look, the 2 weeks, the wild plant up topping can grow like that, wild type, but the control, the treatment is still maintain the succulus. So that's the so far best result we have. So I don't have a summary slides, but I do have an important summary slide like that. So I acknowledge all those people. Then James Strickland was my VP who is retired. Then others, all co-workers with a different group. Then last one on the list, Mac Nielsen, lead my career, carry together all the burdens I put on him. Then also we developed the nicotine demethylase together. With that, I'm happy to entertain some questions. Hey.
I guess I have a quick question. I wonder, in your perspective, what's the most important environmental factor threatening the tobacco production of?
Wow, that's $1 million question on a long story. Then all experts are there here. I'm a little student here. So compare, then a lot. Depends on what you're looking to. Start from day one, germination, fertilization, the temperature to get in, and gradually to the later stage, all will affect it. Then the biggest one, if you're looking to nicotine content, will be the fertilizer. Second is on the drought, on the water. If the root didn't develop big, then the top portion will not develop so much. Last year was the example. But if your root system grow more, then your nicotine level, octanoid level, alkaloid level will be high. If you're regarding TSNA, so many different environment impact. So start from, again, zero seed. If you have less nitrogen, even just the one thing in the leaf, then during curing, same condition, your TSNA will be low. But if you have adverse condition, however, then TSNA probably reach to roof as well. So everything's relative. I know I don't answer your question, but it's rather complicated.
Appreciate it. That means unexpected environmental change is very important to our management.
Yes, that's why we have agronomic group intensively, like for just example, for very low nicotine lines, you plant to different region, then the level of nicotine, the quality will be day and night. So every single trait we developed or every single line developed, we need a match with the appropriate agronomy practice protocol, and also the leaf curing procedure. That would be ideal, but reality will not always happen. So manage to find a happy medium, have the harvestable, one of the best quality you can get with given year. Because 1 year, if it's too much moisture, no matter how well you do, then your quality will not be that great. I'm talking about TSNA level. So yeah, but that not misleading. It's not, doesn't mean cannot do the genetic control. Everything is relative, right? Yeah. So
I just want to say for the more accurate weather forecast, very important.
Great point. Yeah, we closely look into that. Then actually from different perspective, irrigation and fertilization application, pesticide control. Then experts again in this audience. I'm not going to say too much. Thank you.
So in addition to the leaf quality issues that you encounter with it seems that you would likely also have problems with deficient as well. Is that something you run into also?
Yes.
If so, is there a solution to that in biotechnology that's already?
Great point. To answer you, yes, we're doing it. Reach to the level we want, not yet. Because yeah, great point, great thinking. Nicotine is insect-resistant agent, right? That's probably where original tobacco gets its nicotine from. Then if you say extreme case, low alkaloid Burley tobacco, we have some plant only have the mid-rib left, rest all eat up. Not only that, lately we found a rabbit. The plants you plant, turn out 1/3 is gone because the rabbit dig out. So a lot of things we are working on some, but everything is limited, So yeah. Oh, yeah, that's one story. I cut off so many slides. Right now, what we look into is market value, so face value for tobacco itself. Of course, I have a slide like uptake, assimilation, storage, redistribution. All those stories can put together, right? But for us, it's to say how much input fertilizer you put in, how much yield with good quality of yield can come out. That's all we do so far. But we do have the detailed analysis. We even do like amino acid composition afterwards, even in the middle. For example, just give you this non-confidential one gene called the GT2. And in this Maryland, they actively act on, the others not, glutamate, you know what I'm talking about. So we do look in that, but this nitrogen utilization efficiency is different from soybean on the, I think, different from soybean on the weight of those, because we look into the leaf and also manageable leaf quality with the leaf quality, and we use less input. I know I don't answer your question, but that's what we do now. Face value evaluation. Because we're a company, we want to look into solution, you know. My group of people has long time of the postdoc experience. If they deeper, I said, oh, hold on, come back. So yeah, we do that all. That's my job. So yeah.
Did you have one last one for us here?
Well, it's great talk about potential for biotech to alter how we grow plants, the quality of our plants.
Mm-hmm.
So what is the current status of commercial acceptance of biotechnology and limitations of biotechnology for tobacco?
Dr. Barrett, I know somebody will ask me that question. That's a touchy question. Yeah, and in the soybean, corn, you guys know more than me. In tobacco, so far, no GMO material allowed. And also have differentiation between that GMO thing. Transgenic, you have transgenic, you have introgenic, right? I have slides for that. Introgenic, that means the gene come from tobacco per se, just a different position contact, then position. Then that USDA aphids particularly potentially cannot prove as non-transgenic. US only. But for EMS, cross here on the international, all non-GMO. That's why we heavily go to EMS, no problem. Because they said accelerate natural breeding, natural mutation. Gene editing in US is non-GMO. Europe, EU is GMO. And also the other technology have discrepancy too. So now we go safe. We only use the tools to find the function. We go to EMS, mutant, and screening. That's our background selection come to play. It's very important for us, powerful tool to speed up. Yeah. Thank you for your question.
Okay, David, you got one more?
Following up on that, so EMS is good for knocking genes out, but if you want gain of function, you often find it with EMS?
So far, that's our limitation. So not yet, like what I'm talking about the flavor generation in the future, then we hope we can use cisgenic, introgenic, cisgenic. So that will be, you know, be a little bit. Also, it says, I'm going to talk about the philosophical issue. GMO is not absolutely banned from tobacco. If they, for example, if FDA say you have to have 0.4 milligram per gram of nicotine, then you cannot technically achieve based on the general breeding or EMS. But this is probably a wrong example because this is knocked down. Then in the future, you want to survive, you have to accept GMO, right? So I don't know what I do not have a crystal ball, but so I just based on the current regulation and go for it. Yeah. Again, breeding, natural breeding come to big play to get the exotic trait from external. Then you do the breeding through the background selection cleanup. That's a good idea. So we do that. Actually, WZ resistance is acquired from different type of tobacco. That's addition of disease resistant trait.
All right.
Okay.
Well, it's our turn now to present you with a little bit of something, a piece of Kentucky to take back and put on your wall for you.
Well, thank you so
Dummy.
much.
I would like to definitely get Todd and others that have been mentioned today to come on up and get some pictures taken with her to celebrate her achievement and our pride in her being a graduate of our degree program. So let's give her a big round of applause.